So, you would recall that earlier we have outlined various ways or the mechanisms by which enzymes catalyze chemical reactions. Now, one of the unique features of the enzyme's catalytic function is their specificity. In fact, when we distinguish them, when we compare them with a variety of chemically catalyzed reactions or even uncatalyzed reactions, they are distinguished by virtue of their specificity. In the case of chemical catalysts, a single catalyst may catalyze a large variety of similar reactions. Take for example, if you consider nickel as a catalyst, now all the hydrogenation reactions can be basically catalyzed by that. Similarly, the case with colloidal platinum, vanadium pentoxide, or for that matter even acid. I mean, for a large variety of hydrolytic reactions, acids or hydrogen ions can act as a catalytic function. On the other hand, when we talk of enzymes, these functions are highly specific. In most cases, the enzymatic catalytic function is both substrate specific as well as product specific. That means, you need different enzymes to act on different substrates, or to produce a particular product, different enzymes are needed. And there is a significant variety as far as specificity is concerned. All the enzymes are again not specific to the same extent. There is lot of variation in terms of their characteristic features required as a specific catalyst. In many cases, the enzymes may exhibit a very broad specificity, not too strictly specific. A large number of examples which you may be familiar with are group specific enzymes, what we call as group specific enzymes. You can think of a characteristic example of these kind of enzymes as hexokinase. Now, hexokinase, they phosphorylate hexose to a hexose phosphate through, of course, involvement of ATP. A very characteristic enzyme. Now, this enzyme particularly will be able to act upon a large variety of hexose as a substrate, and it can phosphorylate, and is not very strictly specific in that sense, and what we consider usually and call it as group specific. That means it is involved in the transfer of a phosphoryl group from ATP to a given range of substrate. Similarly, even in the case of alcohol and alcohol dehydrogenase, which can also again involve NAD, and it can give you the corresponding aldehyde. Another example of a group specific enzyme whereby a variety of alcohols, particularly primary alcohols, are oxidized to their corresponding aldehyde involving NADH system as a cofactor. So, now these enzymes are often, you will also appreciate, they are often involved in many of the constitutive functions of a living cell, where the cell wants a very broad specificity, and that is why the nature has evolved these group specific enzymes. There are certain enzymes which are highly specific, which exhibit absolute specificity. Now, absolute specificity, when I say, I mean that the enzyme can recognize only one substrate. Same, if you consider the same class of hexokinase, a very closely related enzyme is what we call as glucokinase. Now, this accepts only glucose as a substrate and phosphorylates to glucose 6 phosphate. No other substrate is, no other hexose sugar is acted upon by such an enzyme. So, they show absolute specificity, and the largest probably or the most absolute specificity you can look at even in terms of an enzyme with which some of you may be familiar is like oxidation of beta D glucose to gluconolactone by glucose oxidase, a fungal enzyme which is produced by Aspergillus niger. Now, this acts on not only on D glucose, but it accepts only beta D glucose, and alpha D glucose is just not accepted. If you consider alpha D glucose, now this on this the enzyme which is essentially alpha D glucose, the enzyme, the particular enzyme does not act, or even for that matter, even if you go to another isomer of glucose, say galactose, it also does not act on even galactose, where essentially you have not much difference excepting that these hydroxyl and hydrogen atoms are interchanged. Now, so these are the types of enzymes which are essentially fall in the category of absolute specificity. That is another category. Now, then we have a number of enzymes which are stereochemical, which show stereochemical specificity. That means they recognize the arrangement of atoms at a carbon atom at which a stereoisomerism can occur, and there are large variety of enzymes which recognize. If you just consider the example of, let us say, lipase from various sources, they can exhibit specificity to only one of the isomer, either a L or a D isomer, or there are large number of such examples in the chemical literature, on the biochemical literature. The only enzyme which is known to act on both the stereoisomers are the enzymes, those function is to interconvert L and D isomers, usually called as racemases. Now, for example, L-alanine to D-alanine, Y-alanine racemase can act on either side of the reaction. It is a reversible reaction, and therefore it can act on both on L-alanine as well as D-alanine. This is only one classical example where an enzyme can act on both stereoisomers. Otherwise, most of the enzymes will exhibit stereochemical specificity, and if there is a asymmetric carbon atom, it will only accept one of the isomers and not the other. Now, even the stereochemical specificity can extend to the level that enzyme acting on a non-symmetric molecule and producing one of the isomers as a product. The typical example is your glycerol kinase. Now, glycerol essentially, as you know, CH2OH, CHOH, CH2OH, it is a non-asymmetric product. The carbon atom both are in the CH2OH, they are identical, and but when it acts on the glycerol molecule, it produces L-glycerol 3-phosphate. That means the enzyme transfers again with the action of ATP to ADP. The enzyme transfer phosphoryl group to the third carbon atom, the third carbon atom of the glycerol molecule. Although the 1 and 3 are identical, but still it is able to exhibit the stereochemical specificity in terms of product formation. So, basically, I mean, what we must appreciate is that the variety of specificity that can occur in the case of enzymatic reaction can be very loosely specific in terms of group specific enzymes, absolute specificity with respect to a single substrate. It can be stereochemically specific with respect to substrate or with respect to product, and therefore the enzymes or in summary one can consider that enzyme will exhibit the specificity both with respect to substrate as well as with respect to product.
So in the case of glycerol, when both the CH2OH groups are same, how do we know on which one has it acted? Like it is a symmetric molecule.
Right. It is as I explained earlier, the glycerol molecule is a symmetric molecule. There is no asymmetry in this, but the enzyme glycerol kinase acts only on the.
So how do we know that it is the third or the first?
That you can analyze. You can analyze the product and see where the phosphoryl group has gone.
But it is symmetry. It is symmetrical. How do we know
Pardon?
which is which?
The first and third will be similar.
So how do we differentiate?
No, but that's what I'm saying. First and the third are similar, but the enzyme transfers phosphoryl group only to the third carbon atom.
So how do we know that is the third or the first? How do we know that that particular carbon to which the phosphoryl group has been transferred is the third?
No, that will depend. Now, if you just consider, let us say, a kind of a structural sort of conformation of your glycerol. Now, this is, for example, is a now it will act upon your this is 1, 2, sorry 1, 2 and 3, and this acts only on the 1/3 which is on the left hand side, because that will be exhibited by the planar, because you see we are expressing here our conformation on a planar diagram. When you look at a 3-dimensional space, in the 3-dimensional space the 1 and 3 will not be in the same plane. So essentially the what I was suggesting was that the specificity can have a very large variety, and again one must also appreciate that the specificity of these enzymes in terms of their catalytic action is again attributed to the active sites, as we also saw in the last lecture, where the catalytic function was studied, understood that the catalytic function of the enzymes is made responsible through the role played by active site. Even the specificity also is a result of the conformation of active site. Essentially we also saw that the active site will consider the binding process, that means certain functional groups on the substrate will bind to certain binding sites in the active site, and the active site will also have certain catalytic sites which will interact with the functional residues on the substrate molecule, which have to undergo change, the bonds that have to be broken. So basically the active site must be considered as consisting of two different parts, the binding sites and the functional site. That is one major understanding which we need to. Another factor of stereospecificity is that prediction of stereospecificity by any ground rule is difficult. We are only able to know the specificity only while we measure or while we determine the amount of product formed on a particular substrate. You cannot predict that, say for example in the case of a chemical reaction to take place, you have delta G value, the free energy change of the reaction, which will give you an idea whether the reaction will take place in the forward direction or reverse direction, or there will be equilibrium established. But in the case of stereospecificity, in the case of specificity of enzyme action, the only way to understand the specificity is that we monitor the product formed on the substrate, and then we can say yes, this is specific to this substrate. Otherwise, and of course once we find specificity, then you can propose hypothesis or mechanisms by which the stereospecificity can be explained. But there are no other thermodynamic or kinetic interpretations which can be applied to the specificity. That is another feature. Now to look at some of the critical understanding of the basis for the specificity, as I mentioned that one of the most interesting feature of enzyme specificity is their stereochemical specificity, which probably demands, because when there is a single molecule, the functional groups are different. Active sites also may have different functional groups to bind them, and the specificity can be understood. But to understand the stereochemical specificity, that how enzyme is able to identify the two different stereoisomers, the minimum requirements on the function of an active site was proposed by Ogston sometime in 1948, as early as 1948, and he proposed that at least there must be three sites of different points of interaction on a active site. That means the enzyme active site, if you consider the bottom one is the whole enzyme molecule and the dotted lines represent the active site, and you will recall that I must again remind you that this is not a planar site, it is a three-dimensional structure on the surface of the molecule, enzyme molecule, and essentially not that all the amino acids involved are on these active sites are located in proximity in the amino acid chain. They have been brought in proximity by folding of the enzyme protein, and therefore now if you consider that, let us say a substrate has to undergo reaction which has three functional groups represented by R dash, R double prime, R triple prime. At least it is understood as proposed by Ogston that the enzyme active site also must have at least three different functional residues on the active site which can interact with the substrate molecule.
So these are binding sites, or
no? Now these three interacting sites, let us consider they are the interaction sites. Minimum two of them must be binding sites. Out of the three, at least two on which the substrate molecule can bind to the active site and put the substrate in an orientation, in a very fixed orientation, because at 2 points you are holding the enzyme molecule bound to the substrate in such a way, this binding has to be oriented in such a way that the third functional group on the substrate which has to undergo reaction is brought in vicinity of the catalytic site. That is one minimum requirement for explaining the specificity of enzyme action, that there must be minimum three sites which interact between the enzyme and the substrate. Two of them must be at least binding sites, and the substrate binds on these two sides to the enzyme molecule, whereby it orients the substrate molecule along with the enzyme such that the reactive functional group and the reactive site on the enzyme active site, they come in proximity or in vicinity. And then of course the various mechanisms that we discussed, by which it could be a strain on the bond, it could be the entropic factors, and it could be various covalent interactions or electrostatic interactions, they might play a role to carry out the catalysis. But as far as the binding is concerned, has to be specific in terms of bringing the functional group on which the reaction has to take place, bring in the vicinity of. Now even if, for example, I'll try to explain the same point with a particular example. Take the example of oxidation of a D amino acid using D amino acid oxidase, where you have this amino group undergoes reaction. Now in the molecule, the carboxylic group and the methyl group are bound onto the enzyme active site. Of course, the one which binds methyl group will be hydrophobic in nature, and this will be bound predominantly by electrostatic interactions. Now these two binding brings the catalytic site in close vicinity of the amino group, which is available for reaction, and the catalytic reaction can take place. Now even if now consider the other way, now for this enzyme, even if we say that now this is your D amino acid, now you consider L amino acid, corresponding L amino acid, on which the location of H and amino group will be different. Even if the binding takes place, this amino group cannot come in the vicinity of catalytic site, cannot really approach to catalytic site, and no reaction can occur, absolutely. and that is why the concept of 3 interacting sites fairly explains the position of enzymes catalytic action as far as their specificity with respect to stereoisomers is concerned. Now, of course, the situation in the case of different substrates which can have different functional groups can be much simpler, because the active catalytic sites, the binding sites will bind 2 of the functional groups. The third functional group, which has to undergo reaction, will be only recognized by the catalytic site, and no other functional group will be recognized, and so reaction cannot take place. Now, one of the very early understanding, I mean, this is one of the, I mean, a bit of surprising feature, that as early as 1890, Oxton proposed such a mechanism which puts a limit, just like in the case of rate of a reaction, we have a thermodynamic limit, that if there is a reversible reaction, these are the kind of limits put. Equilibrium constant will be achieved at this point. Similarly, Oxton proposed a limit that minimum there must be 3 points of interaction. Of course, in case of very hardcore specific enzymes, the point of interaction can be more than 3 also. There can be more, I mean, instead of 2 binding sites, there can be 3 binding sites also. That depends on the requirement for the enzyme. Now, as early as in 1890, the Fischer proposed lock and key hypothesis, which explained the enzyme specificity. Of course, you must appreciate that this explanation of the specificity was purely based on certain observations and analysis of the situation, mostly mental and not analytical, because not many analytical facilities were available. And what he suggested was that the enzyme active site and the substrate have a conformation which is analogous to lock and the key. That means the slots on the key are exactly fitting to the site at which it has to interact with the lock. They are exactly fitting, and that is what here, as I have shown, you have the active site which has the sites on which the binding groups available on the substrate. Now, this portion shows the substrate which is analogous to the key. This is the active site is analogous to the lock. Now, the binding sites on the substrate, they exactly come in proximity or bind to the binding sites on the active site of the enzyme molecule, and similarly the reactive group on the substrate again also comes in contact with 2 catalytic sites on the, and then once the reacting group gets entangled with the reaction sites on the active site, the binding is complete and it can exhibit the catalytic function to take place. Now, that was the understanding, and they also very clearly understood at that time that both substrate as well as active site on the enzyme molecule are rigid structures. I mean, there is no question of change. If they are rigid in nature, they have, just like a lock and key, there is no flexibility. If there is slight modification either in the conformation of the key, it will not fit to the lock, and thereby the reactions will not occur.
So, is this true only for a macro fit?
Yes, in fact, the Fischer's lock and key arrangement, as I mentioned earlier, that this was not based on any of the analytical proofs. It was only a conceptual explanation given, because at that time, one thing we must appreciate, that not a very large number of enzyme reactions were understood. Even stereospecificity was not very clearly understood. It was mostly some very common reactions like hydrolysis of sucrose to glucose and fructose. Most of the hydrolytic reactions were understood, and this mechanism proposed only such a macro understanding that the substrate and the enzyme are rigid conformations which are complementary to each other and which fit in very strictly. And for example, I mean, probably at the time of Fischer's and lock and key mechanisms, the most understood enzymatic reaction was hydrolysis of sucrose. by invertase to glucose and fructose, and obviously they would look into variety of other sugars like lactose on which the enzyme would not act, and so they could distinguish between the conformation of the substrate molecule, and obviously much detailed information, but still the surprising thing is that even that concept is not totally invalid today. In many cases it can still explain the whole theory or whole requirements of specificity. Then came, I think, the number of other developments, and particularly the developments with respect to the availability of analytical data on enzyme substrate binding. People were able to make enzyme substrate complexes, their analogues, analyze them by NMR or EPR or variety of analytical physical tools, and they could see, they found that when the enzyme substrate do bind, there is a significant change in the conformation. It remains, the conformation is neither the same what was available in the case of enzyme molecule. So there is something going on, and keeping those data in view, the Coastland proposed a hypothesis as late as in 1958, that what we understand today as Coastland's induced fit model. Now his proposal was a slight modification of the lock and key arrangement. He said that yes, substrate is a rigid structure, rigid conformation with its binding sites, binding groups, and the functional site or reacting groups present at in a particular conformation. But the enzyme active site is not a rigid conformation. It is a floppy conformation, it is a flexible conformation. That means as soon as the substrate comes in the proximity of the enzyme active site, some binding takes place, initial binding takes place, then enzyme the enzyme active site binds on to the substrate molecule. It just encloses, for example, this is a functional group, this is let us say a substrate, the enzyme active site just comes, as soon as it comes proximity to it, it wraps around the substrate molecule, and therefore the in fact he gave an example, as a matter of fact, that the interaction between the substrate and the enzyme is something like a hand and the woolen glove. You wear a woolen glove, your hand is analogous to substrate. Then when you put in a wooden glove, it exactly fits into hand depending upon the conformation of your fingers and all that, everything goes in line, and it sets in which sort of interacts with different fingers, and one can be comfortable and the reaction can proceed. So basically the basic difference was, while the lock and key mechanism or hypothesis proposed both the components to be rigid, in the Coastland's induced fit model it was very clearly understood that the substrate may be rigid, but the enzyme active sites are not rigid conformation. As you can see here, for example, you have two binding sites, the free active site on the free enzyme molecule without interaction with the substrate has a particular configuration where the catalytic sites, binding sites are distantly located. As soon as the binding groups come in contact with the binding site, as they have interacted here, the active site undergo a change in conformation, and these catalytic sites are brought in proximity so as to wrap around the reactive group on the substrate molecule, and essentially then the ultimate conformation of the enzyme substrate complex, which can use one of the mechanism by which catalytic reaction can take place, can be applied, and one can carry out. Now such a mechanism very effectively can explain the high degree of specificity for the enzyme molecules, and now in that case in the case of induced fit hypothesis, one can also appreciate that certain reactions, for example, even if there are certain contaminants in the reaction mixture which are very similar to nature in terms of their conformation to the actual substrate, they will also not be able to interact. For example, in the case of a lock and key mechanism, if there are molecules which are very similar in conformation, they might be able to partly come into interplay with the catalytic site and influence the reaction. Whereas in the case of the induced fit mechanism, once the enzyme active site is totally wrapped around the substrate, now the reaction sites are not at all available for any other contaminant, and therefore there is no chance of any other contaminant present in the substrate molecule to be able to interact with the enzyme molecule. Now, induced fit, one of the most typical example of enzyme catalyzed reaction which was responsible to sort of elucidate the induced fit model was the hexose kinase. I mean, as I mentioned earlier, the phosphorylation of dihexose at the expense of ATP to hexose 6 phosphate, and now in the absence of hexose, the ATP is hydrolyzed extremely slowly, because ATP can hydrolyze a spontaneous reaction. Hydrolysis of ATP is a spontaneous reaction, but in the absence of hexose, it will hydrolyze only very slowly. When the hexose is present in the proximity of the substrate, in the proximity of the enzyme will bring both hexose and ATP along with the binding sites, and then it can flop around the two molecules, and the reaction can take place at a very fast speed. In fact, the induced fit mechanism can also explain very clearly the bimolecular reactions, in fact, which was difficult to understand as far as the lock and key arrangement was. Lock and key arrangement was very easily able to explain the unimolecular reactions, but in the case of where two substrates are involved, as in the case of hexose and hexose phosphorylation, the role of hexose and ATP both participating at the binding site and for the reaction to occur could be explained using induced fit mechanism. Now, if you consider all the three explanations that we have considered today, like Ogston's 3-site interaction model, lock and key mechanism of Fischer, or induced fit hypothesis of Coastland, while they talk about the specificity, but they do not link specificity by any means to the catalytic function, because that means they consider specificity totally in isolation. They only say that yes, after the all these interactions have taken place, some of the reaction mechanisms responsible for catalytic function will take place and the reaction is catalyzed. None of them is able to provide a suitable explanation for the catalytic function. Now, as a matter of fact, we also know, when we discussed our the various ways in which the enzymes are able to catalyze reactions, we also understood that binding of the enzyme to the substrate was a very important feature in the acceleration of the reaction rate, and of course binding of the substrate to the enzyme involves some expenditure of energy. There is a binding energy involved in it, which ultimately gives you an advantage in reduction in activation energy, of course, but there is initial energy. Now, it is also presumed that this binding energy is not totally a wasteful energy for the enzyme reactions. A part of this binding energy also contributes to specificity, and therefore if you look at the effect of strains that are developed, I mean, in one of the sort of hypothesis which is called as a strain, that means at the stage of binding, as we understood in the case of lock and key mechanism, that they are rigid, but we might assume that, sorry, we might assume that the active site is almost complementary. Active site is almost complementary to the substrate molecule, but is not exactly complementary. I mean, a situation which is slightly closer to lock and key mechanism, not absolutely in absolute terms, that they are complementary, but they are not exactly complementary, and therefore the binding when the substrate and the enzyme's active sites are brought in proximity and the binding takes place between the substrate and the enzyme active site, it brings in a strain. You see that the active site here, unlike the lock and key mechanism, is not exactly complementary. This is slightly this slot is slightly longer, and therefore while the binding takes place, it makes a sort of a strain onto the reactive group, and it is analogous to the mechanism which we proposed for catalytic function, that means strain mechanism, and which contributes to reduction in the enthalpic component of the free energy of activation. And during this interaction of the enzyme active site with the substrate, automatically the strain produced onto the reactive group as a result of binding process is responsible for specificity as well as part catalytic function. This strain developed may contribute towards the reduction in activation energy, but it is a result of binding process, and this binding process essentially brings in specificity, and the nature of the binding sites or the active site on the enzyme molecule is slightly complementary or of the similar type, but not exactly complementary, and a complementarity comes only after binding has taken place, which results in the distortion of the reacting group, and then the complementary comes in. So that is called the strain effect. Strain effect, which also can be consumed, which can take into account both catalytic function as well as specificity.
So where is it taking into account the catalytic function, reduction in the enthalpy?
Yeah, in the strain effects, as we discussed in the time of when we were talking about the ways in which the enzyme reduces energy of activation, the strain on the reacting group was one of the mechanisms. So here, because the active site is not exactly complementary, it forces after the substrate approaches the binding sites, it forces the reacting groups to develop a strain, so as to sort of bring in complementary to the catalytic sites, and then only these catalytic sites can combine. Otherwise, see, for example, if it was totally complementary without this strain, then the reacting group will come down somewhere here, and no reaction can take place. Only when a strain is developed, the reaction takes place, and the reaction takes place with a suitable degree of specificity introduced into it.
So that means strain developed is due to the catalytic function.
Due to the binding sites, because the binding has to take place, and the binding process requires a strain onto the substrate molecule, so that it comes in proximity of the catalytic sites. If strain is not developed, it will remain as a binding site, but not as the active site. The enzyme will not be able to provide the catalytic function. Just like in the case of other mechanisms or other hypotheses proposed for specificity, while we are able to explain that yes, binding will take place, the functional groups will come in proximity to the reacting groups, but it does not explain that after that whether the reaction will take place or not take place. Okay, just coming in proximity could provide you an effect what we discussed also, like high substrate concentration in the proximity of the active site, but does not explain you how the energy of activation is reduced. Here in this case, this also explains the energy of activation is reduced in terms of its enthalpic contributions. Now, another thing which is very well related to this strain mechanism is a mechanism what we understand as transition state stabilization. Transition state stabilization. Now here in the case of, the effect is almost similar, very similar to what we call a strain effect, but what is understood is that while the substrate enzyme binding takes place at the active site, there are still some unfavorable interactions at the active site with the substrate. That means the enzyme and the substrate molecule in the vicinity of the active site is still not very comfortable in terms of its interactions. There are many other unfavorable interactions available at the active site, and therefore the role of the enzyme is also in the sense that these interactions tend to distort the substrate in such a way so that it favors the formation of a complex which is analogous to structure to transition state. The presence of certain unfavorable interactions at the active site for the substrate when the binding has taken place tends to distort the substrate molecule in the direction so that the enzyme substrate complex tends to become complementary to the transition state, and essentially if you
Complementary to the transition state.
yes, similar to transition state, not as a matter of fact, if you remember in our first characteristic features of catalysis, we saw that the enzymes the transition state is complementary to enzyme substrate complex and not the substrate. The enzyme is complementary to substrate, is complementary to enzyme substrate complex, the enzyme active site, and not to the substrate itself. And therefore the role of the binding of substrate molecule to the enzyme at the binding site doesn't the substrate doesn't remain very comfortable. There are many unfavorable interactions still present there, and they distort the substrate molecule so that this enzyme substrate binding approaches towards that of transition complex.
So could you explain this please?
You see, the first step is binding of the binding sites onto the active site. Now once that takes place, still the catalytic sites have not approached to the functional groups which are likely to undergo reaction. But in addition to the binding sites, there are many unfavorable interactions present between the substrate and the active site, because active site, although we are considering, let us say 3 different functional groups, but in addition to that on the active site there will be many more amino acid residues which will have in the proximity, and they might provide some kind of unfavorable interactions. Now these interactions tend to distort the substrate, and this distortion of the substrate is favored towards reaching to the transition state. Now this, in fact, the basic role is played by the unfavorable interactions present at the active site. Now in another way, it is basically to say same thing in a way that you are developing a strain onto the substrate molecule so that the substrate molecule reacting groups are coming in proximity to the catalytic site. But what in this hypothesis of the transition state stabilization, what is stated is that the unfavorable interactions available at the active site for the substrate favors the distortion of the substrate in such a way that the enzyme substrate the final enzyme substrate complex approaches in structure to that of transition state. So basically the energy required for transition state to reach a transition state is already reduced, because partly you have already taken up that path. That is what happens in strain development also. Part of the energy required for energy of activation is already met by strain onto the substrate molecule. So these are some of the major mechanisms that have been proposed for explaining the specificity of the enzyme action. Now basically, as I mentioned earlier, it is the overall rate of reaction in terms of rate of product formation that indicates whether the enzyme is specific to a substrate or not. What is the order of specificity? to what extent it is specific, what are the impurities which can interfere in the specificity. They are all related to the results of the reaction, that is the rate of formation of product. Now the specificity also has a very significant role in terms of the industrial application of enzymes. We must also appreciate that while in the case of, let us say, the catalytic function, we understand that if the rate of reaction is increased, its usefulness as an industrial catalyst is also very high, because higher the rate of reaction, you can very beneficially use it for industrial purposes. But what is the role of specificity? Now the specificity, in fact, if you look at, has both negative as well as positive role. Positive role in the sense that you can restrict the formation of any of the byproducts. Even if your feed substrate, the feed stream substrate is impure to some extent, whatever reaction will take place is only which can be catalyzed specifically by that enzyme which you are providing, and no other reaction will take place. And even the side products formed by action of water, usually most of the organic chemists consider water for organic chemical reactions as a culprit, because it can lead to a variety of side products. So all the side products can be absent, which can make our life comfortable as far as the purification of the product is concerned. That is one of the major advantages. But the major disadvantage or drawback from the industrial application point of view is that the specificity requires a single enzyme to be produced for each reaction. So basically, as an engineer, you will appreciate that the benefits of scale of operation for the enzyme production is lost. That means many of the enzymes cannot be produced on a very massive scale, because the requirement will be very, only for single reaction they will be used, and therefore for a single reaction the requirement will not be very large. So obviously you have to produce them on a very small scale, and producing on a small scale will mean the high cost of production, and that is why enzymes shall still remain to be by and large expensive catalyst compared to chemical catalyst. For example, in the case of chemical catalyst, Raney's nickel, it is a catalyst used for all hydrogenation reactions in the organic chemical field, and you can produce them in bulk quantities, and the cost of production can be reasonably low. On the other hand, if you have to produce, let us say, glucose oxidase, which acts only on beta D glucose, not even alpha D glucose or any other hexose, its requirement will be only for oxidation of glucose only, and which will restrict its application, and therefore the scale of operation. So essentially the specificity must be looked at from both the angle, and it is in fact in many cases today people are applying the developments in molecular biology, particularly in sense of protein engineering, to modify the substrate specificity. Both wherever we want the absence of side products to make it more specific, as well as where we want broad specificity so that the same enzyme can be applied to a large variety of substrates, we are trying to broaden the specificity through protein engineering method.